This experiment is to determine the endpoint of the neutralization reaction between sodium hydroxide and hydrochloric acid using phenolphthalein as indicator. The equation of the reaction is shown on the screen. 1 mole of sodium hydroxide and 1 mole of hydrogen chloride react to form 1 mole of sodium chloride and 1 mole of water. To begin this experiment, we will assemble our materials first. Materials needed for the experiment are a burette with a capacity of 30 or 50 cubic centimeters, a conical flask with a capacity of 100 cubic centimeters, 2 beakers each with a capacity of 100 cubic centimeters, a pipette with a capacity of 25 or 30 cubic centimeters with a pipette filler, a stirring rod which is optional, a small filter plastic funnel of about 4 centimeters in diameter, a burette stand and clamp, and a white tile or card, which is also optional. The chemicals needed are 100 cubic centimeters of sodium hydroxide solution with a molar concentration of 0.1 mole per liter in a labeled and stoppered bottle, 100 cubic centimeters of dilute hydrochloric acid in a labeled and stoppered bottle, and phenolphthalein indicator solution or an alternative in a small dropper bottle. Precautionary measures to be taken are: use protective equipment such as a lab coat, eye goggles, and hand gloves. Make sure the stopcock at the bottom of the burette is not stiff, blocked, or leaking, as this will make the experiment unsuccessful. Label the beakers before pouring in the various chemicals to avoid mistakes or contamination. Collect some of the chemicals in the individual beakers that have been properly labeled. Using a pipette, add 25 milliliters of the hydrochloric acid solution from the beaker to a conical flask. The hydrochloric acid solution is of unknown concentration. Add 2 drops of phenolphthalein indicator to the conical flask containing hydrochloric acid. Shake up the content. Add some sodium hydroxide from the beaker containing sodium hydroxide to the burette. The sodium hydroxide solution has a known concentration of 0.1 mole per liter. Place the white tile or card on the base of the burette stand. This enables any color changes to be easily visible. Then place the content of the conical flask on the white tile. Open the burette so that the sodium hydroxide solution is released directly into the conical flask. Add the sodium hydroxide solution dropwise or in small amounts at a time and swirl the conical flask as you do so. You would notice that at each drop, the solution turns pink but goes colorless after swirling. Keep adding the sodium hydroxide solution and swirling the flask. Turn off the burette as soon as the pink color remains and doesn't disappear. You have reached the end point when the pink color does not disappear. How much sodium hydroxide was used to neutralize the acid? Record the amount of sodium hydroxide used from the burette mark. Repeat this experiment at least 3 times to get results within a 0.2 cubic centimeter margin of each other. Take an average of these results to use in calculation of unknown concentration of the hydrochloric acid. Here are some real-world applications of titration. In the food manufacturing industry, titration is an analytical technique used to determine the quantity of an ingredient in a food sample. For example, it can be used to discover the amount of salt, fat, water, vitamins, sugar, and so on in a food product. It is used in biological labs to determine the concentration of certain chemicals in blood and urine samples. It is an important process used in the pharmaceutical industry to ensure quality control. It is a key mechanism in wastewater analysis, winemaking, and in environmental studies to quantify the degree of contamination in natural rainwater or snow.